Welcome everyone to day 2 of our Stereo-CITE version 1.1 webinar series. I'm Luo Ping from the Stereo-CITE Application Support Center. Today I'm going to present you getting started with Stereo-CITE version 1.1 experiments. In my section, it covers 3 main parts, including how Stereo-CITE works, the experimental workflow, and keynotes for sequencing. Ready to dive in? Let's get started. And since the dawn of microscopy, with Robert Hooke's first glimpse of the cell in 1665, science has driven by a fundamental quest to see life structure with ever greater clarity and finer details. This journey to understand the intricate architecture of biology has now led us to one of the most transformative technology in life science, spatial technology. Nowadays, life science are transitioning from single-dimension development to multi-dimensional models. Single-layer data often proves insufficient for a comprehensive biological insights, driving the development of integrated multi-omics methodologies. Now, Stereo-CITE is proud to introduce you Stereo-CITE as one of the examples of this technological advancement in spatial multi-omics. And now let's have a look how it actually works. The Stereo-CITE applies cellular indexing of transcriptomes and epitopes by sequencing. And this allows simultaneous detection of whole transcriptome and high-plex protein on the same section. Quality-based capture with ADT is applied to identify each protein of interest. Here is a diagram illustrating the antibody structure. Each protein of interest is detected by antibodies with specific conjugated oligonucleotides, which is also called antibody-derived tag, ADT. Each protein of interest is assigned to a unique antibody barcode, which allows the protein identification in sequencing. The commercial antibodies compatible with Stereo-CITE is Stereo-CITE antibodies from BioLegend. You may refer to Stereo-CITE recommended commercial antibody list and in-house validated individual antibody list from our official website to look up the antibody information for experiments. In brief, user may use antibody cocktail or select individual primary antibodies for the Stereo-CITE experiment. Antibody cocktail is a commercially available premix of primary antibodies, and they are high-plex protein panels. Like we also call it universal cocktails. And these kind of panels, they include more than 100 antibodies and their isotype controls. And there's also a small panel which specifically targets any cell types of interest. And here is an example is the human TBNK cocktail. It only includes 9 antibodies, primary antibodies, but it's specifically for the principal lineage immunocells cell types. And user may also select individual primary antibodies and mix them together for the Stereo-CITE experiments based on their research interests. Now, let's have a general overview of this experimental workflow for our Stereo-CITE version 1.1. Briefly, this workflow consists of 5 parts: sample preparation, antibody titration, promorphization and optimization, proteome transcriptomes, library preparation sequencing, and visualization. The first step is always prepare your fresh present sample. After the tissue embedding, take sufficient amounts of your sections and extract the RNA for the ring value detection. Normally for our Stereo-CITE version 1.1 experiment, you should have a sample with a ring value no less than 4. And you should also have an H&E staining on the glass slide to assess your tissue morphology. Make sure there is no deformation, fragmentation, or ice crystals. And with your chosen primary antibodies of interest, you should perform an antibody titration on the tissues. The primary this pilot study aims to find an optimal antibody concentration for your proteome transcriptome experiment. If you're using a high-plex universal cocktail, you may skip this step. And for another very critical pilot experiment is the promorphization optimization, and we also call it P-chip test because we will use the Stereo-CITE chip P to find an optimal promorphization time for your tissue before your proteotranscriptomics experiments. With all these pilot studies, we then proceed to the proteome transcriptomics workflow. We apply the optimal concentration, antibody concentration based on your titration result, and we apply the promorphization time based on the optimization result. And we can then proceed to the this experiment, we will have the reverse transcription, and then we will collect all the cDNA and ADT and separate them through purification. After rounds of the amplification and purification, you will have your cDNA and ADT products, and also will proceed to the libraries for and ready for the sequencing. After the sequencing, you get a sequencing data, and you analyze, process and analyze your data with all the Stereo-Mix offline software. And this part will be introduced by my colleagues afterwards. Now let's break down each part of this experiment. The first part is the antibody titration, and you can refer to the operation details in the Appendix 2 from our Stereo-CITE proteome transcriptomics set user manual. And briefly, first you have your cryo section ready. You section out, mount it, and fix it on the glass slide. And then you will apply a blocking buffer incubated with our primary antibodies and secondary antibodies, and assess the best concentration through imaging. And here is the result of antibody titration. And normally for antibody titration, we will have 4 sections, including 1 negative control with no antibody added. And with the 3 concentration tested, the gold standard for selecting the most optimal one is the lowest concentration with sufficient fluorescence across your tissue. And so in the example here is the 1 to 250 gives the best, because it has the signals across the tissue, but it also gives you the lowest concentration. And for the Stereo-CITE universal cocktail, because it is pretty expensive as well, and we also tested an optimal concentration internally in R&D. So we have also listed in our validated list in the document previously I mentioned. And so you may skip this antibody titration for cocktail if you're using. And another important part of the experiment is the permobilization optimization. And this will require to use our specific Stereo-CITE permobilization set and a Stereo-CITE P-chip. And we have the user manual respectively for our operation details. In brief, we will also need to cryosex our section first, and we mount it on our P-chip and fix it, and this takes up around 1.5 hours. And then we will block it and have a mock antibody incubation, which means we will not use any antibodies in this part. Because our key part in this, the key aim of the experiment is to find the optimal permobilization time for your tissue. And then we will apply the glycerol mounting, tissue wash and fixation before the permobilization time. We have different time permobilization time set, having reverse transcription, tissue removal, and we will proceed to the imaging to get the result. And here is an example of our permobilization optimization. And the gold standard to finding the best time for your permobilization is the chip with the highest or with the strongest signals with more or finer details in the tissue. And 12-minute one I'm showing here is actually giving the best result. And this is a mouse pancreas actually. You can see that it gives you the highest signals across the tissue, and also give you more details. And you see the 18 and 24 minutes, they're actually not giving you very even. They're giving you uneven signals, and not the details of the structure of the tissues can be seen. And the 6 minutes is really not many. It's pretty dim in terms of the signals. And as I mentioned, to minimize the antibody usage, no antibodies are required for permobilization optimization. With our pre-knowledge gained from the pilot experiment, we then smoothly move to proteome transcriptomics. And we have the specific reagent set and the user manual for your operations. And firstly, you need to section them, and you mount them on our Stereo-CITE T-chip. And then you fix them in the PFA, 4% PFA. And then you'll block them with blocking buffer to minimize all the false signals caused by the non-specific binding between the antibodies and antigens, or between the ADT and the probes. And then you will incubate your antibody, both primary and secondary. It will also need to stain your cell with DAPI, which is gonna stain the cell nucleus. We have the Debye staining and imaging because we wanted to have we need to have an image registration to our final gene matrix after the sequencing. And so this part is very important. And for imaging, you're not just gonna image. Besides Debye imaging, we also will have the immunofluorescence imaging, which is gonna detect whether your protein has successfully bind to your target, the antibody. So basically, the signal is emitted from the secondary antibody fluorophore. And then you will have the tissue wash to remove all the non-specific binding antibodies, and you'll have a fixation before the permeabilization. Permeabilize the tissue with the time you optimized previously, and you have a 2 to 5 hours reverse transcription, and you have your cDNA ADT after the release. And we separate them through the base purification, and we have a couple rounds of amplification before we proceed to the library preparation. With our cDNA and ADT purified, we will proceed to library preparation, and we use another kit. And we will have a library preparation user manual for your reference. And for cDNA, in general, we will have 3 hours for a cDNA library, and for ADT, we just normally have 1 hour for a library. And you can tell we will have an amplification and purification, and then we have a size selection, and then after that we have another amplification for cDNA library. And for ADT, we just basically shoot through amplification, and then we have the purification. And for a cDNA library, we normally have a fragment distribution with the main peak ranging at 200 to 600 base pair. And for ADT, we have the library fragment distribution ranging at with the main peak at 200 to 260 base pair. Now, here is the main part also for the sequencing. For the Stereo-CITE libraries, it can be sequenced either by DNB-seq T400 or DNB-seq T7. These two machines are compatible. And for T400 sequencing, you need to sequence the cDNA and ADT libraries separately. And if you're doing a T7 sequencing, you will need to separate them. You will need to sequence them together. And so since different strategy was applied, the sequencing settings are different as well. For RIG1, both libraries are the same. 50 base pair cycle 26 to 40 will be set into the dark reactions. But for cDNA, the read-through length will be 100 BP, and for ADT, it will be 36 BP for the cycle, and it will set the cycle 1 to 21 for dark reaction. And for T7, since they are pulled together and sequenced together, the read1 and read2 parameters will be the same. And here's a side note: if you have one same ADT library and you sequence first separately at T400, and you get the sequencing data, and then later you have some top-up sequencing with the T7, and you sequence together with the cDNA. That means two different strategies with the used for the same sample. And be aware of that. You cannot use the two sequencing data together for analysis, since they have different sequencing strategy applied for the ADT library. Now, you may expect the total reads out for your library. And for cDNA, we expect to have 1.5 billion to 2.5 billion reads for your sample. And this also depends on the tissue area and your analysis requirements. And we have a formula for your reference. And for ADT, if you apply a universal cocktail, which includes more than 100 antibody used for your detection, we expect to have 1 billion reads for your sequencing. And we're using a small panel antibody, no matter if you use your primary antibodies of chosen or using a small panel. For 10 plex, we expect to have 300 million reads. For 30 plex, we have 400 million reads for expectation. And 50 plex, we will expect to have 500 million reads. And here are the major equipment will be used for our Stereo-CITE experiment, which include cryostat, fluorescent microscope, Slitrail, and thermocycler. And we have specific requirements for the fluorescent microscope. For those of you who are new to our product, we need to remind you that you need to assess whether your microscope of chosen is compatible with our Stereo-CITE products. And you could seek assistance from your local FAS and your microscope engineer for microscope assessment. We have this document on the online on our online official website. It is called Stereo-CITE Image Requirements and Guidelines. We will produce. We will need you to perform a pilot test to see whether your microscope can really image our Stereo-CITE chip. And then here's a key imaging system requirements for your microscope. They're the same as other Stereo-CITE products, but there is one key thing I would like to highlight for Stereo-CITE experiment is that make sure your microscope has the filter or the channels specifically for the your secondary antibody. So because we have the secondary antibody, you're going to assess whether your protein has successfully bounded. And if you do not have that specific channels or filters, then you probably will not see any signals in your imaging. So here's a keynote for you guys. And for other reagents, equipment, and consumables required for Stereo-CITE solution version 1.1, please visit our official website under the document category and refer to the experiment preparation checklist. We have all the required materials listed out for you. You can visit the Thermic's global website for more product information and instructions. Thank you for listening. And upcoming next, let's welcome my colleague Hao, introducing how to process and analyze your Stereo-CITE data.
The Stereo-CITE version 1.1 platform has seen significant updates. It maintains nanoscale resolution across a centimeter scale field of view, allowing for the capture of more genes and over 100 proteins at the same time. This results in a high-quality unbiased spatial data for both the whole transcriptome and the target protein. To analyze this amazing data, we have released the SW version 8.2 and the StereoMap version 4.2. Today, I will show you how this tool transfers raw data into clear, actionable biological insights. Here is our agenda for today. We will start with the core principles of Stereo-CITE technology. Then we will dive into our offline software SW and the StereoMap. Following that, I will walk you through some practical scenarios. And we will finish with some additional information and the resource. Okay, let's begin with the core principle behind the Stereo-CITE. Here is the capture mechanisms of the Stereo-CITE version 1.1 platform. Our chip contains lots of capture spots with a poly-T sequence. These spots bind the poly-A tails of mRNA molecules and the poly-A tails of ADT molecules. So this allows the co-detection of the unbiased transcriptome and the target protein on the same chip. Here is an overview of Stereo-CITE workflow following the completion of wet lab experiments such as sample preparation, blocking, imaging, and library construction and the sequence. The next steps involve data analysis and visualization. The data analysis requires three types of raw data. First, a chip mask file, which are associated with spatial locations with coordinates CIDs. Image file that can be used to identify the tissue and the cellular information. And additionally, RNA FASTQ file, it includes both CIDs and RNA sequence data, while the ADT FASTQ file provide protein-related information. Once all of these files are prepared, we integrate and analyze the data using our offline software SW and the StereoMap. SW is primarily responsible for the computational analysis, like alignment of Stereo-CITE sequence data. For the still map, it enables spatial visualization and it can perform image quality control and segmentation. Okay, next let me give you more details about SW first. SW or still-seq analysis workflow is a comprehensive tool for spatial omics data analysis. The core module for SW is SW count. It provides a standard pipeline for spatial data analysis. In addition, SW also includes several complementary pipelines. Like a MakeRough, it builds a genome index for alignment. Sometimes if you are unsure about your genome annotation file, you can use SW check GTF to verify its format and the content of your gene annotation files. Together, these tools simplifies spatial omics data analysis. Today, due to the time constraints, we will mainly focus on 3 key pipelines: SW count, SW MakeRough, and SW realign. Okay, before analyzing your spatial omics data, a genome index must be prepared for the species of interest. The genome sequence file and the gene annotation file can be downloaded from some public websites, such as Assemble and NCBI. Here is an example script showing SW MakeRough command. You need to improve a gene sequence file, gene annotation file, and then specify the output directory. SW will automatically build a genome index. And for Stereo-CITE data, which also includes proteomics, so you need an additional file, a protein panel. This maps 15 base pair barcode sequence in the ADD data to their corresponding protein. This panel can be downloaded from our official website, so this link here. And once you have both the transcriptome reference and the protein panel, you should list their path in a reference.csv file. And now your reference setup is completed. Okay, now let's look at the core principle, SW count. As I mentioned, it is a standard workflow for processing your spatial data. The required inputs are reference.csv file that we have prepared before, the triple mask file, the RNA FastQ file, and the ADT FastQ file. Also, we need input microscope image file. One key thing I would like to highlight here is that you must correctly specify the kit version and the sequence type for when you analyze the Stereo-CITE data. For example, the sequence type for transcriptome may be 15 plus 100 base pair, while that for ATT data could ATT could be 15 plus 36 base pair. And sometimes they can be the same, both are 15 plus 100 base pair. So please be careful about be careful when you input this information, and in providing incorrect information here may lead to an error or incorrect results. When the SW count runs successfully, it generates a comprehensive set of output files in the auth directory. This includes alignment files, registered imagery, future expression metrics, secondary analysis results like clustering, gene differential expression analysis results, and also there are 2 important files. One is the HTM report, which provide the basic statistics of your spatial data analysis results, and also visualization.gz file that you will use in StillMap. And sometimes the automatic results on SW count need refinement, especially for image registration or segmentation. This is where SW realign comes in. If your, if your image fail quality control, or you have manually processed it in still map, you can use the realign pipeline. As showing on the right, you need to provide the series number of your chip, the path of the original SW count results, and the compressed image file from SW from a still map that contains your manual adjustments. And the SW realign pipeline will then reanalyze your data. Okay, this was about the main features of SW. Now let's move to the still map, our tool for visualization and exploration. Here is the main interface of still map version 4.2. It has 3 modules: Visual Explore, Image Processing, and the tools. To start exploring your data, you click Visual Explore and navigate to your file system. Select the stereo file from the decompressed visualization.txt archive, and SW will automatically load the results from the SW. By default, and you will see a heat map of spatial gene expression. In the future menu, you can select specific gene to see their spatial distribution. You can adjust the display layers and customize the visualization effects. A key fact for our product is the ability to change the bin size from a very high resolution bin 1 to bin 200 for a broad view. And you can also switch to a cell bin view if cell segmentation were performed. All the same visualization operations are also available for the proteomics data. You can, as you can see here, you can view protein expression heat map across the whole tissue. You can adjust the bin size, and you can explore just as you do with your transcriptome data. For clustering results, only bin 20, bin 15, and cell bin views are available by default. If you need other bin size, you can generate them using the SW reanalyze pipeline. And in the right panel, you can manually annotate cluster and adjust their color for better visualization. The same operation are also available for proteomics data, and you can check the cluster distribution in the across the tissue. Like here, we can see there are 5 clusters. Yeah. And for the still map, one of the powerful feature is the ability to view protein and its corresponding gene expression side by side. For example, here we can see the protein CD22 on the left short screen, and here on the right is its corresponding gene CD22, and you can check whether their distribution are expected. Beyond the visualization, StillMap also enables the downstream analysis. The Lasso selection tools allows you to manually outline regions of interest based on the tissue morphology, gene expression, cluster results, and you can annotate this area for deeper investigation. Again, the same operations are also available for plotting data. As you can, as showing here, you can lasso the region based on the plotting expression pattern, and also the cluster results for plotting. After your annotating your regions of interest, you can explore their spatial coordinates and load them into SW reanalyze lasso to produce spatial future expression matrix for selected regions. And you may also input other GeoJSON file to conduct differential gene expression analysis. As showing here, you can compare the labeled one and the surrounding regions. Okay, this concludes the Visual Explorer module. Now let's look at imagery processing module, which you used when SW automatic processing needs manual adjustment. The imagery processing module involves a step-by-step process. First, you need to input imagery you want to adjust and provide relevant information. Once that's done, click Next button here. The second step is imagery alignment. Here, you will manually align the imagery with the gene expression matrix from SW count. StillMap provides tools for flipping, rotating, and moving the imagery to improve alignment. There are also normalization, contrast adjustment tools to fine-tune the imagery for better results. And here is an example of fine alignment. In addition, there is an alternative method, future point registration. This way, use the check line detected from the imagery and the chip mask line based on predefined rules. It doesn't require the spatial future expression matrix generated by SW, which may save your time, especially when working with complex tissue areas. However, I should mention here, this feature is only available for some product lines. So, also, your tissue image must pass the image quality control first. If you are unsure whether this feature is available for your chip or not, you can read our StillMap menu in details for more information. The third step is image segmentation. First is tissue segmentation. You will segment tissue by identified regions. You can either edit the current mask or create a new one using the lasso and the eraser tools. Also, you can input a mask from a third-party tools like QPath. Next, you will segment individual cells. This is similar to tissue segmentation, where you will edit the mask to define cell boundaries. In addition, in this version of StillMap, we also introduced a watershed algorithm to help users to identify cells. Also, you can use the third-party tools like QPath, CellPulse, and send the results as a mask to the image to the StillMap. After completing image registration, tissue segmentation, and the cell segmentation, click Explore Image Processing Records to generate a .gz file. This file can be used as input for SW realign for further analysis. Okay, now let's look at final module tools. Currently, the tools module includes one function, image quality control, which check whether your microscope images are suitable for automatic analysis in SW. We plan to add more tools in the future version. Okay, now let's look at a few examples for the user's scenario. In first scenarios, automatic processing. In this case, the user perform automatic analysis using SW. They first perform image quality control, then SW count pipeline. Once the results are generated, they can use StillMap for visualization and further exploration. The second scenario, the user work with the image that requires manual processing. After confirm that the image has passed the quality control, but before the sequence data is available, the user can perform future point registration, as I mentioned before, and manually adjustment the image like tissue segmentation, cell segmentation. And once the sequence data is ready, they can input them into SW for analysis and then visualize the data in the still map. The third one involves images that fail quality control or where automatic image processing for SW count is not perfect enough. In this case, using image processing module in the still map for manual adjustment. And after modifying the image, they re-engine it into SW real-line pipeline for reanalysis, and finally visualize the data in still map. And sometimes still map and SW are combined for more in-depth exploration. Now let's delve into how analyze and explore your results with still map. As I mentioned before, SW allows you to explore gene and protein expression pattern and their cluster results at both square and the cell bin level. Here, by overlaying the tissue imagery, you can visually assess cluster locations. And also, you can explore the protein and its corresponding gene expression at the same time. And based on these insights, you can identify the regions that need further exploration. You can select regions based on tissue image, gene expression pattern, or cluster results. And for instance, you may be interested in these two regions you can select using the lasso tools, annotating them, and explore them into SW. As showing here, there are example scripts for lasso and differential gene expression analysis. To analyze gene expression between these two areas, you can select. You can use the lasso to select these two regions, and input relative .geojson file into SW reanalyze differential pipeline. It will help you explore how gene expression varies across the different area. And here is the example results for differential gene expression analysis. Okay, that includes our overview of software and its applications. Let me finish you with some additional important information. These slides provide basic information of our software. For SW and the StillMap, both of them suppose the product line, Stereo-CITE version 1.1. And for SW, it's a Stereo-Omics tools for Stereo-CITE bioinformatic analysis workflow, while for Stereo-Map version 4.2 is a tool for visualization and image processing. And here showing their requirements and operating system. And for the final slide, it's also showing the compatibility details for the software and the various kit versions. You can find more information by visiting our official website. Okay, thank you for your listening.